Our next presentation is from one of our senior PhD students. So we have the pleasure to listen to Jeff Okamoto right now, and then we'll go right into the admissions part. So thanks, Jeff. Go ahead.
Hey everyone, I'm Jeff. I'm a PhD student here, and I'm going to be talking about some of my recent work on integrating transcriptome-wide association studies and colocalization analysis. So quick show of hands, has anyone ever heard of either of those before? One? Okay, that's more than I thought. Okay. So I'm going to spend the first few minutes filling you guys in on what exactly colocalization and transcriptome-wide association studies are. Then I'll talk about my method and talk about a quick real data application. So the broad research question that we're interested in here is how can we study the genetic mechanisms of complex traits. You can think of a complex trait as something like your standing height or maybe like a disease status. So one of the most popular ways that researchers have done this recently is called a genome-wide association study, or GWAS. So these allow us to see basically the SNP level underpinnings of a complex trait. So what's a SNP? Basically, it's just a single substitution of a nucleotide at some place in your genome. And the idea of a GWAS is basically to perform a simple linear regression of your complex trait levels on your minor allele count at that SNP. And if you do that basically hundreds of thousands to millions of times across your genome, you end up with a plot like this, where you have basically an association for every SNP in your genome, and you have the association strength here on the y-axis and the location on the x-axis, and we call this a Manhattan plot. So the next step is basically linking these genetic associations to the genes that they influence, which we call putative causal genes here, and this is actually pretty challenging. So traditionally, researchers have relied on our prior biological knowledge of genes in proximity to those GWAS loci. So we have the same Manhattan plot here as the last slide, and I've revealed to you that it's a GWAS for serum testosterone levels in females. And you can now see that they've annotated the top GWAS hits based on that previous biological knowledge. So fairly recently, a new class of methods has begun to emerge called mechanism-aware PCG implication methods. And the idea of these methods is to take what we call multi-omics data and integrate that to identify PCGs and reveal their underlying mechanisms. So some examples of common data types, multi-omics data types that we often integrate are transcriptomics or gene expression and proteomics. And some popular methods that we use to do this are called colocalization analysis and transcriptome-wide association studies, or TWAS. So what's a colocalization analysis? So the goal here is going to be to identify, or to determine whether genetic variants that are causal for a molecular phenotype, so gene expression, for example, and we call these variants QTLs, overlap with those that are causal for your complex trait, so your GWAS trait. So here I have a little cartoon. We have SNP-X, which is causal for both your gene expression of some gene, gene A, and your complex trait. So this represents a colocalization. So this is actually a fairly complicated task in practice, and I'll show you why with these two cases. So case one on the left, we have our true colocalization. Meanwhile, case two on the right, we have separate QTLs and GWAS hits, so we don't have a colocalization. So it turns out that if SNP-X here is perfectly correlated, or in genetics lingo we say in perfect linkage disequilibrium, or LD, with SNP-Y, then these two cases are going to be indistinguishable. So in response to this problem, researchers have developed Bayesian methods to try to resolve this case if SNP-X is in weak or moderate LD with SNP-Y. And basically, the idea is that you can quantify the uncertainty in the presence of colocalization through what's called a gene-level colocalization probability. So that was one way to link genes to your GWAS trait. Now I'm going to talk about another way, which is called TWAS. So TWAS is a form of what's called instrumental variables, or IV analysis, which is designed to use observational data to test for causal relationships from some exposure, in this case gene expression, to some outcome, in this case your complex trait. So we're interested in whether or not this red arrow on this graph exists. So this form of analysis comes actually with some pretty strong assumptions because we're using observational data. One of them is randomization of your genetic variants. One is the relevance of your instruments or your genetic variants to your molecular trait, so just your QTL strength. And then the final one, which is often the most difficult to validate, is the exclusion restriction, just being that your SNPs can't be causal for your complex trait through any pathway other than the exposure that you're considering, in this case, our gene expression. So in practice, the way that you do a TWAS is you have some expression reference panel on the right here that you use to train a prediction model. Then you use those prediction weights in a separate GWAS data set to predict your expression across the transcriptome. You end up with your transcriptome-wide set of predicted expression. Then you correlate all of those predicted expressions with your GWAS trait. So you end up usually with like a transcriptome-wide set of association statistics, like a z-score. Just to recap a little bit here, colocalization analysis was a way to probabilistically quantify our overlap between causal QTLs and GWAS hits, often returning a colocalization probability for each gene. And TWAS was another way to link our genes to our trait. This way tests an association between your predicted expression and your complex trait, and you often get a z-score as output for each gene. Recent work has actually found that these two types of analyses can be complementary when applied to the same data. In particular, they don't always implicate the same set of genes as causal. And there are certain underlying biological factors that can actually explain this. For example, if you have a strong colocalization but weak TWAS signal, this can indicate the presence of what's called horizontal pleiotropy. So we have here on the left what horizontal pleiotropy looks like, where you don't actually have that arrow from your gene expression to your complex trait, so this is not a causal gene. Meanwhile, we're looking for cases like on the right where you have vertical pleiotropy, where you do have that effect. And on the other hand, if you have a strong TWAS signal but weak colocalization, this can indicate what's called an LD hitchhiking effect. So in this example, you have SNP-X, your QTL that you're using. That's an LD with some direct effect SNP, SNP-Y. And basically, this is inducing a false or spurious TWAS association. And you can see in this case, you don't actually have colocalization. So going off of that, this previous work offered a strategy to try to reduce these spurious TWAS results. And the idea here was going to be to basically filter your TWAS results using a colocalization probability threshold. So the idea here, graphically, is going to be to superimpose colocalization evidence, and hopefully that'll get rid of cases of spurious, where you don't have a true causal effect from your gene to your trait, and hopefully you'll just be left with your true scenarios where there is an effect. So the major drawback of this approach is it's pretty ad hoc, and you lose your uncertainty quantification in your PCG implications. Okay, so now we're going to get into our methods overview, and our more focused research question now is going to be how can we take our evidence from these two types of analyses, TWAS and colocalization, and probabilistically integrate them to implicate our PCGs. So we're going to start here with a model to motivate ourselves. So the top equation is for our gene expression of our target gene, our candidate gene. And this is going to be a function of the genotype matrix G and our true eQTL effects vector beta E. Our bottom equation Y is for our complex trait, our GWAS trait. And this is a function of our true gene to trait effect, which is what we're interested in, gamma, as well as this extra G beta Y term, which represents the pleiotropic effects, which are not mediated by our gene expression. So previous methods have actually also considered a similar equation, but these impose additional assumptions to try to identify that gamma term. For example, they assume that one method assumes that the two beta vectors, beta E and beta Y, are uncorrelated, and another assumes that your pleiotropic effects are constant across variants. So in practice, these actually aren't very reasonable assumptions. They're often violated in real data. So to try to get around making these assumptions, we're going to focus more on testing whether gamma equals 0 rather than estimating it. And as sort of a launching point, we're going to note, based on this equation, that our causal eQTLs are going to have to be co-localized with GWAS hits based on this model. And basically, to show you that, just sub in the E equation into the Y equation, and you can see that genetic variants with non-zero beta E's are also going to have a GWAS effect. So our method, which we're going to call integration of TWAS and co-localization, or INTACT, basically is going to incorporate that observation into an empirical Bayes framework to implicate PCGs. So a quick overview here. We're going to first form a Bayes factor from our TWAS z-score to represent marginal likelihood. Then we're going to form an empirical Bayes prior using our co-localization evidence. So that's going to take the form pi f of p colloc, where p colloc is our co-localization probability. We're going to estimate pi as a unconstrained TWAS prior, and the function f is going to satisfy two properties, the first being that it's monotonically increasing with our co-localization probability, and the second being that it's thresholded at some value t, just to make sure that if we have a very strong spurious TWAS signal, we don't want that to overwhelm a very small co-localization probability. And then at the end of the day, we can use Bayes' rule to form our posterior. Okay, so summing up here again, our goal was to estimate a posterior probability of causality, essentially, for each gene. We used an empirical Bayes framework to do this. That involved converting a TWAS z-score to a Bayes factor, and then we also formed an empirical Bayes prior based on our co-localization evidence. Okay, so now I don't have time to show you how well our method works in simulation, but I'll jump into a quick real data application. So for this application, we're going to look at 4 different GWAS traits. So we have serum urate, IGF-1, and testosterone separately for males and females. And the reason why we chose these traits is because we actually have a lot of biological knowledge ahead of time about these traits, so we can actually use these to see how well our method is working. So these traits can be further broken down into subpathways, which is in the second column to the left. And each pathway is annotated with a number of core genes, or genes that we just know are relevant to these complex traits from our prior biological knowledge. So that's annotated to the right of the pathway name. And then the middle column is going to be the number of core genes that we have implicated based on the proximity plus knowledge approach. So proximity plus knowledge is just basically the approach that I talked about on one of the first slides, where basically you implicate any gene that has a GWAS hit basically in proximity to its coding region in your genome. So to the right of that, we have the number of genes implicated by INTACT, and we're integrating multi-tissue gene expression data for this implementation. And then finally on the right, we have the overlap of the core genes implicated by both approaches. So what I really want to point out here is 2 things. First of all, we have substantial overlap between the number of between the genes implicated by each approach. You can see that on the right. And then also, there are several cases where INTACT implicates core genes that we miss using the standard proximity plus knowledge approach. So we're going to conclude here that INTACT is a complementary approach to the standard. Okay, so just wrapping up here. So INTACT is basically a new method to explicitly link genes to complex traits using the transcriptome. The main idea here was to protect against LD hitchhiking effects by constraining our TWAS results using co-localization data. And then we just saw that we were able to complement the proximity plus knowledge approach. And some extensions that we've been working on recently are adapting INTACT to consider additional molecular phenotypes, so protein levels, for example, in addition to our expression. And then also to focus more on trying to estimate those effects rather than just testing for them. So I'd like to acknowledge my advisor, as well as our co-authors on this work. And if you're interested, I encourage you to check out our paper and our package. Thanks for listening.